Good afternoon, ladies and gentlemen. I am delighted to welcome you to the second meeting of the Scientific Forum Age Open Science. I would like to thank the audience for joining us in such a wonderful historic scenery, the patio of the Palacio Anaya. During this session, we will address a matter that with no doubt concerns all of us, longevity. I would also like to give a warm welcome to the audience that is following us on streaming. To begin, I call on Eugenio Santos, Director of the Cancer Research Center and the Foundation of Cancer Research of the Universidad de Salamanca, to take the floor.
Okay, good afternoon, everyone. It is a real pleasure and a privilege for me to be here today in this Palacio de Anaya doing the introduction of Dr. Giovanna Mallucci and her participation in this Age Open Science Forum that we are celebrating under this very original format in different places in Salamanca today and tomorrow. I was told to be very brief, so I will just say that Giovanna works at the Dementia Research Institute in the Cambridge University in the UK. This is a world-leading institution housing scientists who work to find new ways to diagnose, to treat, to prevent, to care for people with dementia. Very shortly, Dr. Mallucci's research focuses on understanding the mechanisms of neurodegeneration. In particular, the central theme of her work is the identification of common pathways occurring across the wide spectrum of all the pathological disorders related with neurodegeneration. As a measure of the quality of her research, I may mention that Giovanna is a fellow of the Academy of Medical Sciences and is also a recipient in 2021 of the very prestigious Protankim Award for research on Alzheimer's and related disorders. Today, Dr. Mallucci comes to talk to us about a topic which is of concern to many of us, particularly to people like me who are starting to have white hair on our heads. And the title of her presentation is going to be How to Boost Resilience in Neurons to Treat Neurodegeneration. Please, Giovanna, the floor is yours.
Thank you. It's very loud. Thank you, Eugenio. And Juan Carlos asked me to say a few words in Spanish. So, buenos días a todos. Gracias de la invitación en esta bellísima ciudad de Salamanca. Okay, but the rest is in English. Okay. So, I'm actually now, I've left the Dementia Research Institute for a new post in Altos Laboratories. Can I have my first slide? Oops. Oh, no, sorry. Okay, that's my slide for today. So I've just started with Altos, but the idea is the same, the pursuit of understanding of mechanisms and new ways to treat neurodegeneration. So for all of you here, neurodegenerative diseases are a group of diseases going from the very common, Alzheimer's, to the very rare, like prion diseases. But collectively, these are a global problem, which is reaching disastrous proportions. So it's already the commonest cause of death in the developed world, but as the whole world ages, and the China and India and the developing world ages, it's the impact of dementia and the fact that it's untreatable, completely untreatable, is not to be underestimated. So all these diseases, although they are all different, they all have similar basics, which is that these are, you're looking at neurons, brain cells, and the sparks between them are the contacts or the synapses between them. And these contacts are where you make your memories, but also how you keep the brain cells alive. So all of these processes have in common the loss of synaptic connections, so the loss of fine function of the brain cells, but also the gradual loss. When these are lost, you lose the neurons. So you have two processes that happen. The decline in synapses that comes over time, and the accumulation of specific misfolded proteins for each disorders. And one of the real reasons that the treatment has failed repeatedly with these diseases is it's all been focused on the specific misfolded protein. And this in itself, to prevent the buildup of these proteins, is not enough on itself. So what we have to do is look for the common processes. I'm not getting in my next. Where's he gone? This isn't moving. So yes, so these combine to make brain cells die. But really, what we have to think about are the common mechanisms underlying the loss of synapses and the loss of brain cells across all of these disorders, so maybe we can find common cures and not focus on the specific. So it's the opposite of precision medicine. This is really what we call boosting resilience. And so if I change the curve, if you can see that when I do my clinics, in hospital, and my patients come to me at about this point, they've lost 70% of their synapses, they've lost early memory, and then they progress to losing brain cells very quickly, and that's the stage of institutionalization and death, and it's irreversible. But if you change the shape of that curve, you can see that 60% of synapse loss is now going to happen when someone is 85. And then the progression to cell death will be when they're 100. So already you have completely changed the experience and the quality of that disorder and the progression of disease in a way that is really very meaningful. If I could hold my patients at the level of synapse loss when they arrive in clinic, then this would be a real progress. Yeah, and this is the concept of resilience. So we've been working on both sides of these curves, on synapse number decline and misfolded protein accommodation, to find ways of making brain cells stronger. So how do we study the mechanisms? We've used a model of disease that is actually a rare disease in humans, but is actually a real disease of mice. So these mice actually have neurodegenerative disease, which is exactly the same as the human condition. So they have all the biochemical, pathological, and clinical features of the human disorder. So instead of the mouse models of Alzheimer's, which mice don't get Alzheimer's disease, but they do get prion disease. So this is a way of studying the relationship between the protein aggregates, okay, which are the clumps, the amyloid plaques, and the death of brain cells. This is an organ of memory, the hippocampus, and all the holes you can see is where the brain cells have died and degenerated, with memory loss and then hospitalization. So the prion mice let us study the relationship, and those protein aggregates that you were looking at are basically the progression of what we call misfolding of proteins, which is the common process in these disorders. And the normal protein, which is correctly functional, misfolds and accumulates. And the best way to describe this is a normally folded functioning protein, and that is a misfolded aggregating protein. And you can see that this is the same protein, but now it doesn't work, and it's sitting in the brain causing a problem. And so this is what's at the heart of all these disorders. What this does is it induces a cellular, or what we discovered initially was that actually if you take away the normal fold during disease, the normal folded protein, instead of all the mice dying at 12 weeks, the mice lived forever. They lived their whole lifespan just by removing this misfolding reaction. And this reversed spongiosis, and what was really important about this is what you can see here is this is the progression of disease from the left to the right. There's no pointer, but it doesn't matter. From left to the right. So you can see that you lose all the brain cells on the top and all the holes come. Whereas if you take out the substrate for protein misfolding, you protect against that neuronal loss. And you have restoration of memory, you have restoration of lifelong survival, and you can do that both with genetically engineered knockout or with viruses that will downregulate the protein. So several ways of doing it. But the key message is that early neurodegeneration is reversible. And if this is true for prion disease, this is true for other neurodegenerative disorders. So how do we access these key pathways that will allow us to understand how to protect brain cells against this? So what we noticed, we studied these mice during their disease, and we noticed that the real milestone effect is a loss of synaptic proteins. So while disease is progressing, suddenly about 2/3 or 3/4 of the way through the disease course, all synaptic proteins are down. And this is the key point for loss of memory and motivational tasks and for behavioral change, so you can measure these changes. And it's followed by brain cell death. Okay, so the loss of synaptic proteins, as I said to you at the beginning, is so key for keeping brain cells alive. And why is it happening? Well, remember the protein misfolding. When this is happening in cells, it sparks a response, a protective response called the unfolded protein response. So this change is something the cells don't like. And what the cell does to respond to the change is it does various things. It refolds the deck chairs, but it also stops making new proteins transiently through this pathway. So PERK is activated to activate EIF2 alpha, and this stops new proteins being made. So that's a transient block to help the cell deal with the misfolded protein stress. And this is rapidly deactivated to restore protein production in cells under normal conditions. What we discovered was that this here is showing you the misfolded proteins that's happening during the brain. And actually what you're getting is a very sustained and consistent high level of signaling through this pathway that results in the halving of protein synthesis rates in sick brain cells, in old brains, that is sustained. So instead of that reaction lasting for a few minutes, it goes on for days and weeks. And the effect of that is fatal. Okay, so the brain cells die. So in order to prove this, we modified this pathway genetically by putting viruses into the brain that would then prevent the phosphorylation, the activation of this EIF2 molecule. And we did it either by putting in the phosphatase that dephosphorylates the EIF2, or by blocking the buildup altogether. And then we used a drug to control for this. So the experiment is all the mice have the same level of that protein misfolding in their brains and the same accumulation, but just with these two here, we've now put a virus in with a genetic modification that's going to stop the unfolded protein response being activated. And the result of that is that protein synthesis rates are restored, that all the neurophysiological function is restored and memory is restored, and the synapse number is restored. And this is not by touching the protein at all. We've just simply stopped the EIF2 molecule getting activated. But importantly, you've got neuroprotection. So instead of having all the degenerated neurons, these mice are completely protected, and they live an extended life, healthy, with good memory and good function. There you go. So our hypothesis was now that forget about the upstream protein, forget about the amyloid, target this pathway, boost the resilience in the neurons, and you will get neuroprotection. So this was an important genetic proof of principle, but can we do it with drugs? And the answer is yes. So this molecule was made by GSK as a cancer drug. It's an inhibitor of PERK. So we put it into our mice. And here, these are mice with the prion disease. And we give the drug at 2 time points, both relevant for disease. So that blue bubble is early memory loss, and the pink bubble is when the mice have a more advanced disease that is progressing to neurodegeneration and death. So if I show you, will this work? No. I think you need to, do you mind printing? Yeah. So you can see here, this should work. Okay. Can you press, please? Sorry, you're going to see videos that hopefully, just press the bar. Is it not working? Okay, let's do the next one. I was going to show you some miraculous cures. Yes, there, down. Okay, it's going. So you can see a healthy mouse that's been treated is doing his burrowing, whereas that is a sick mouse, and he really isn't. It's not functioning. His brain is not functioning. And they have the same amount of disease and misfolded protein. Would you mind doing this again? I think you just have to press the video bar. Oh, maybe I can do it now. Okay. Sorry. Okay. Next one. This one. No, I'm not touching. There. Okay. Fantastic. Next one. We'll just do one more. Next one, just the video. So again, you have two mice with identical amount of disease. One's been treated, one hasn't. You can see the paralyzed back legs. And we'll just do this one as well. Because this is cure. This never happened before. This is a small molecule that's cured neurodegeneration. That's treated mice. That mouse is not moving. He falls over. He has this abnormal posture. I think I'll move on. Yeah, you're going to see the last one. And you'll see how extreme this is. This mouse is so sick he can't even correct his posture, whereas the healthy mouse, you cannot put him on his back. So this is a very big deal. You know, there's never been a small molecule that you can give to a mouse to prevent neurodegenerative disease. And it's doing purely by restoring protein synthesis rates. And that in itself protects. You can see how protected the brain cell layer is. So this got a lot of press and a lot of interest from the general public, from researchers. And it's because all of these diseases have this pathway activated in them. Alzheimer's, Parkinson's, frontotemporal dementia. So theoretically, doing this could treat all of these diseases. And in fact, we found another model of dementia. This is a frontotemporal dementia mouse. And again, the GSK drug cured that. So the bottom line is that there's been an explosion in this field of proteostasis, looking for small molecules that can change this pathway, with a lot of new drug development and new drug discovery. But what we did in our lab, my PhD student, Mark Halliday, instead of making new drugs, decided to try and find drugs that already exist that might act on the concept of repurposing. And long story short, he found two drugs, that both in prion disease mice. So trazodone is a licensed, safe antidepressant, and DBM is in clinical trials. But these are as effective as the experimental compounds. And they work both in prion disease and in frontotemporal disease mice. They work in Parkinson's models. They work across the board of mouse models of neurodegeneration. And the key is here that this is already safely prescribed. And it's safe in the elderly. And we need to try it. This is just to show you. OK, sorry. So let me show you. This is some data from a student in the lab. What you see in green is new protein synthesis in real time in brain slices, and that's all in the synapses. That's in a normal mouse brain slice. This is in prion disease. You can see that the whole of this area, this whole neuropil, the synapse area, has almost no protein synthesis, and 2 weeks of trazodone treatment puts it all back on. So this is restoring new protein synthesis in brains in real time, and it completely changes the translatome as well. So what we're trying to do is take this into humans, so translating not just at the level of protein synthesis, but into humans. And what we're doing is imaging this pathway, because I showed you the pathology is present in all the brains. We're imaging this pathway to measure protein synthesis using PET imaging. And here we're using a single labeled amino acid to measure protein synthesis in real time in humans. And this is data that is preliminary, but these are 10 control patients, and that's the mean signal in real time of new protein synthesis in human brains. And you can see the change in signal with Alzheimer's. So more goes to less. That's quite significant. And if you do a map, you can see the hippocampus, the temporal lobes, and the occipital lobes all have significantly lower protein synthesis. So, of course, the question is, can trazodone reverse this? And can it reverse it and make people better? So we're working towards doing clinical trials for dementia with trazodone. And obviously, this would lay the pathway also for other drug development in this field. So I think I hope I've shown you that modifying a generic pathway here, UPR activation, can change the impact on neuronal loss and on survival. I just want to show you quickly some data on another side of this story, which is the loss of synapses. So one of the questions, sorry, there we go, yeah. So as I showed you earlier, the first thing that happens, even before the misfolded proteins build up, is that synapses start to die. And we need to understand why they're dying so early. And I just want to show you how we count and how we look at synapses. So this is an electron micrograph of synapses in the hippocampus, where you have 3 parts. You have the part with the vesicles, which has got the neurotransmitter, the cleft, and then the postsynaptic density, that's a whole synapse. Now, synapse numbers, as I said, they decline very, very early, way before. The red is where protein synthesis rates start to change with the UPR, but the synapse rate starts to decline much, much earlier than that. So you can see that in the context of lost synapses, activation of the UPR is super stressful, because then, of course, you don't make your synaptic proteins. So why do synapses die early, and can we boost synaptic resilience? So synapse number is maintained by a process of plasticity, which is structural plasticity, which is a balance between pruning, when you cut them down, and regeneration. And our hypothesis was that regeneration is deficient and fails very early in these disorders. And to test this, we went to the phenomenon of hibernation. So hibernators, to save energy in cold weather, do all sorts of things to metabolism, but the brain uses so much energy that these animals disconnect their synapses. So a normal hibernator, and this is well known, will disconnect its synapses, and you can see in the middle, so you've lost the density and all the vesicles are pooling, so there's no transmission across a synapse in a hibernating animal. There's very flat brain waves. And then on rewarming, it's reformed. And this process is important because these animals, when they wake up, need to remember where their nuts are. They need to remember where they've hidden their food. So this restoration of synapses must be controlled in a way that's meaningful. So what we did, we speculate. We first tested the capacity for this structural plasticity on cooling in mice, because lab mice are not hibernators. And in fact, they do have this phenomenon. And this is important itself, because this shows that a common mechanism that's used in extreme environments is actually probably true to most mammals. And so we showed that the capacity for structural plasticity on cooling is retained in wild-type mice. So now we did the easy experiment. We looked at the models of disease. This is an Alzheimer's model on the bottom, because they do lose synapse in our prion mouse. And we cooled and rewarmed them very early in disease, when they keep their plasticity. Then just a little bit later, they lose this plastic response. So they're now not able to restore their synapse. So plasticity is defective. The regeneration is defective. And that's in both Alzheimer's and in prion mice. And so importantly, you can see how early this happens. It's the first step, and it's what makes you super vulnerable. And this happens not just with disease, but with aging. So they become superimposed on each other. So what is the mechanism? Well, on cooling and hibernation, you have a response that we know they're protective, especially to the brain, but you have a downregulation of protein synthesis and metabolism, except for a group of proteins called the cold shock proteins. And one of these is called RNA binding motif protein 3, or RBM3, which is induced on cooling. And in the interest of time, I'm just going to show you that by raising RBM3, either just by cooling mice with disease, so these have all got prion disease, as before, and just by raising the temperature and seeing what happens with time, you'll see that cooling alone protects against synapse loss. So the top left, in the white box, is prion disease at 7 weeks, and it's got very few synapses, and the others have been cooled, and their synapses are protected. So it protects it. And that restores behavior, it restores memory, and it is fundamentally neuroprotective. So these mice haven't had any drugs, they haven't had any genetic modification, they've been in the fridge, literally, twice early in their disease. And that gives you this effect on cooling, and this effect on longevity and survival. And it's the outlier here that tells you quite how protective this potentially is. Now, we can mimic the effects of cooling by expressing RBM3 alone, and this is the mouse experiment. We just put in the RBM3, or we knock it down, and again, we see fantastic protection on synapses just by expressing RBM3 during disease, in Alzheimer's, in prion, and amazing neuroprotection and extended lifespan. Come on. Okay. So that's a very simplified flow diagram of cooling increases RBM3 and is neuroprotective. And clearly the interest here is whether we can do this for treatment of dementia. And in fact, throughout neuroprotection. But obviously you don't want to make people cold, but you would like to increase RBM3. So although cooling is used, but you can't possibly, it requires an intensive care bed. You get pneumonia, you get blood clots. So really, we want to drug this pathway rather than put people in the fridge. And we can drug this pathway. I'm not going to go into this slide, but it is absolutely druggable. And we can increase RBM3. And the relevance in humans is, as I said, this is used. It's used for neonatal brain injury. It's used in head injury and whatever. And there are people who voluntarily get hypothermic. And these guys, this is a pond in London called Hampstead Heath Ponds. These people swim every day. They swim in iced water. They get hypothermic. They phoned me up and they said, can we work with you? So we measured RBM3 in human blood for the first time, actually, and in the babies. And both in the babies and in humans, and in swimmers, the cold induces this protein. So you need two things when you're translating from a mouse model to a human. You need to know what you're doing in the mouse is relevant for humans, and it may well be. So we're hoping next to be able to induce RBM3 for therapy of many brain conditions, but including dementia, to increase resilience. So that's these are two common processes. There are others, but I hope I've shown you that a non-precision effect of boosting resilience at various levels and targeting the things that neurons need to, or brain cells need to stay functioning and alive, particularly their synapses, is a way of offsetting neuronal loss. And if you can delay that, then really you will have changed the clinical experience of dementia completely. And science has one of its 125 unanswered questions. To what extent can we stave off age-related diseases like Alzheimer's? And a 5 to 10 year delay would improve old age for millions, and it would have enormous economic benefit. And I just thought I would put that up in Spanish as well, just to end. So I want to stop there. I hope I haven't talked for too long. This is my fantastic team. Mark discovered, this is our first meal post-lockdown, everyone has this slide, but Mark discovered trazodone. Hector did all the amazing, he's from Spain, he's from Alicante. He did all the beautiful work with trazodone, changing the translatome, and everybody else has done various things. And I just want to show you another slide. This is my lab in the Baltic Sea in Copenhagen in November, getting some cold shock therapy. And I need to just thank my collaborators. Do I get up? I go back. I give this to Manuel. That's for you. Thank you.
Thank you so much, Giovanna, for sharing your investigations. I think we'll all go to the sea to give a quick swim. I would now like to call on Eugenio Santos to introduce the next speaker.
Sí. Okay. Como en el caso de Giovanna antes, pues tengo que decir que es un placer y un honor pues el estar aquí para introducir la figura de Manuel Serrano, de Manuel Serrano, que es de los científicos actualmente trabajando en un laboratorio en España, probablemente el más reconocido internacionalmente en el momento actual. Manuel Serrano, como todos probablemente sabéis, trabaja en el Instituto de la Recerca Biomédica, en el IRB en Barcelona. Estaba antes trabajando en el CNIO en Madrid. Muy rápidamente, la lista de su producción científica es realmente impresionante. Quizá muy rápidamente mencionar algunos de sus trabajos originales, de sus descubrimientos originales. El descubrimiento y la caracterización del oncogens del tumor supresor P16 fue un trabajo seminal continuación de eso pues, fue el establecimiento del concepto de oncogene-induced senescence, senescencia inducida por oncogenes, que realmente abrió un área totalmente nueva en la oncología molecular, etcétera, etcétera, los supermice, los superratones resistentes a cáncer, etcétera, etcétera, no me extiendo. Simplemente continuando en esa línea de trabajo, pues más recientemente lo que ha hecho Manolo es demostrar que la senescencia celular es crítica no solamente para cáncer, también para otros procesos patológicos, como fibrosis quística, etcétera, etcétera, y también, sobre todo, que la senescencia celular está implicada en muchos procesos fisiológicos, como por ejemplo la remodelación tisular que ocurre durante el desarrollo embrional. Así que en este aspecto creo que hoy nos va a hacer un resumen, un update de sus avances y de su desarrollo de trabajos en el campo de la senescencia celular y el cellular reprogramming, la reprogramación celular. Incluso probablemente nos contará algo sobre las nuevas aproximaciones terapéuticas, tratando de abolir el desarrollo pues, de la gran cantidad de enfermedades que están asociadas con la senescencia y el envejecimiento. Así que nada, Manolo, aquí tienes.
Bueno, muchas gracias por la invitación. Un placer compartir este rato con Giovanna Mallucci y con Eugenio Santos. Voy a ir rápido porque vamos un poco retrasados, así que me voy a saltar algunas diapositivas que no son importantes. Voy a hablar sobre senescencia celular. A ver, no sé dónde tengo que apuntar, quizás. Sí. Bueno, la senescencia celular, primero, ¿qué es una célula senescente? Pasa a la siguiente, por favor. La senescencia es una respuesta al daño celular. Cuando las células reciben una gran cantidad de daño, a veces son capaces de morir, pero muchas veces no mueren. Se mantienen en un estado que se llama senescencia celular, que está aquí indicado. Bueno, ¿y por qué son importantes estas células? Qué curioso. Está al revés. Vale. Bueno, estas células son muy importantes porque cambian el entorno tisular. Por un lado, se detienen, dejan de proliferar, y por otro lado, secretan muchos factores al medio, al tejido, y lo cambian, lo alteran. Ahí está su importancia, en que cambian el entorno, envejecen el tejido, por decirlo de alguna manera. Y este tejido envejece por todos estos factores y se producen los procesos de fibrosis, inflamación y inmunosupresión, que son casi comunes a todas las enfermedades degenerativas que ocurren con el envejecimiento. Esto sería un esquema, una idealización. Imaginaos que esto es un tejido normal. En todas las células, en todos los tejidos, incluso cuando somos jóvenes, hay un porcentaje de células senescentes, porque siempre hay daños celulares estocásticos debidos al sol, debidos a infecciones, virus. Todo esto produce células senescentes que generalmente el sistema inmune las elimina eficientemente. Se estima que las células senescentes en personas jóvenes tienen una duración de aproximadamente una semana. Sin embargo, a medida que envejecemos, el sistema inmune deja de ser eficaz. No elimina estas células con la misma rapidez y la consecuencia es que se acumulan estas células senescentes. En un tejido de una persona de 80 años puede haber un 5% de células que son senescentes. Tener un 5% de células no funcionales no es grave, no pasa nada por eso en general. Lo grave es que estas células están secretando todos estos factores que están alterando la funcionalidad del tejido y están induciendo esa fibrosis y la inmunosupresión. También es importante que producen esa inmunosupresión. Desde hace unos 7 años, bueno, otra cosa muy importante es que hace, sí, como 7, 10 años, se ha ido descubriendo que muchas enfermedades asociadas a la edad están acompañadas de una gran cantidad de células senescentes, hasta el 50%. Esto lo que quiere decir es que todo nuestro cuerpo acumula células senescentes de una manera progresiva y en poca cantidad, dispersas, pero cuando hay un tejido que está enfermando, ahí se acumulan en gran cantidad, un 50%. Esto también nos hace que podemos ver en muchas enfermedades como un envejecimiento acelerado de ese órgano. Hoy día la fibrosis pulmonar se considera un ejemplo de envejecimiento acelerado del pulmón y el fallo renal crónico como un envejecimiento acelerado del riñón. El resto de los tejidos están bien, pero ese concretamente tiene un envejecimiento acelerado. La infección por COVID, hoy se sabe que en el pulmón produce una gran cantidad de células senescentes y que eso va a ser responsable de lo que se llama el COVID persistente o largo, porque ese pulmón ha quedado envejecido prematuramente. Bueno, esto es una lista de muchas enfermedades en las que se ha asociado una gran cantidad de células senescentes a estas enfermedades. Como veis aquí, hay muchas enfermedades, como he mencionado, en el pulmón, en el hígado, la aterosclerosis, bueno, etcétera. También las enfermedades como Alzheimer, Parkinson, están asociadas con muchas células senescentes de lo que se llama la glía. Son todas las células de soporte que son muy influyentes en las neuronas. Bueno, una cosa que ha sido importante en los últimos 5 años o más o menos ha sido el descubrimiento de fármacos que matan a las células senescentes. Esto es lo que se llaman fármacos senolíticos. Estos fármacos senolíticos han cambiado la manera de aproximarse a muchas de esas enfermedades y hay algunos que están teniendo mucho éxito de momento en modelos animales, pero también hay ensayos clínicos, como voy a mencionar en un momento. Bueno, los fármacos estos senolíticos lo que hacen es intentar ir de esa situación a esta situación o a aquella, intentar reducir la cantidad de células senescentes para que no haya esa fibrosis ni todos esos factores inflamatorios. Estos son una lista de fármacos senolíticos. Algunos de ellos están aprobados para uso en humanos, como el que está arriba del todo, que es el dasatinib, que es uno de los más populares, aunque el que mejor funciona es este que se llama navitoclax, que no está aprobado, pero que ha pasado todas las fases reguladoras de fase 1, fase 2, fase 3. Al final no se aprobó por una toxicidad, pero ahora todos estos fármacos se están reconsiderando para volverse a utilizar en determinadas enfermedades. Como el dasatinib está aprobado, este es otro, los glicósidos cardíacos, en el que participamos en su descubrimiento, y también están aprobados para uso para corazón. La consecuencia de todo esto ha sido que hay ya muchos ensayos clínicos, o algunos, están aquí listados, aunque no sé si lo podéis leer, casi todos con este fármaco, el dasatinib. Ha habido algunos que han fracasado en la fase 1, pero también ha habido otros que han tenido éxito, y concretamente el que se está haciendo en fibrosis pulmonar pues es un estudio muy prometedor. Así que esto es algo que deriva de la investigación básica y que muy rápidamente ha pasado a los estudios en animales y luego en ensayos clínicos. Y con esto pues voy a enseñar un experimento y voy a terminar en 2 minutos, en el que lo que intentamos es demostrar que las células senescentes per se son capaces de generar la enfermedad. Porque todo lo que os he enseñado antes, las enfermedades son producidas por el COVID, o son producidas por el tabaco, o producidas por el alcohol. o por una infección, pero lo que queremos es producir la enfermedad solo con las células senescentes. Entonces, lo que hemos hecho ha sido utilizar células senescentes de pulmón, que las tenemos en el laboratorio, y se las introducimos en el pulmón a ratones. Y miramos a ver si esos ratones desarrollan la enfermedad. El pulmón de los ratones no lo estamos dañando con nada, no lo estamos irradiando, no le estamos haciendo nada, simplemente poniéndole células. Si estas células son normales, desaparecen. Pero estas células que metemos son senescentes y aquí están marcadas como anidan en el pulmón de estos ratones. Esto es a los 2 días. Si ahora dejamos que transcurra el tiempo, vemos, estos son 3 horas, 48 horas, y estas células siguen ahí, siguen, y a las 2 semanas ya podéis ver cómo han generado la enfermedad. Veis la diferencia que hay entre este tejido y este. Esto es como debería de ser un pulmón normal, lleno de aire, con unas paredes muy finas, y ahí es el pulmón fibrótico. Ese pulmón ya no puede respirar y no es funcional. Y esto es una enfermedad que se ha generado a partir de estas pocas células que van generando esta enfermedad progresiva y que va extendiéndose y extendiéndose hasta que mata al animal. Esto es una demostración de que es el agente causal de la enfermedad, son las células senescentes. Si las células que introducimos no son senescentes, no pasa absolutamente nada. Y en este modelo las podemos tratar a los animales con las drogas senolíticas, con los fármacos senolíticos. Aquí hay un ejemplo de ratones a los que se les ha puesto células no senescentes, no pasa nada. Se trata con nabitoclax y no pasa nada, evidentemente, pero a los que se han introducido células senescentes se ha producido la enfermedad, aquí lo veis, la diferencia, y ahora si a estos ratones se les trata con el fármaco senolítico, se revierte. Y con esto voy a terminar porque este es el mensaje principal. Este era un ejemplo que quería enseñar también, haciendo reconstrucción por imagen de los pulmones de ratones. Y aquí se ve las zonas de fibrosis, está aquí y allí. Y este ratón, este concreto, al cabo de 18 días, se ha podido eliminar la fibrosis parcialmente. Y bueno, pues con esto termino y os doy las gracias por vuestra atención y muchas gracias a todos.
Muchísimas gracias, Manuel. Si no te importa, te quedas porque vamos a escuchar algunas de las preguntas. Giovanna, would you mind joining us on stage as we will now listen to some of the questions that have been made by the audience? Giovanna, I would like to start with you. Would you let us know if, do you believe that at any point we will succeed at eliminating diseases like Alzheimer's, for example?
So I think it's a very interesting and important question. I think I have no doubt that we will change the clinical course of these diseases. And this is the really key question. Because what you have to do is keep, you have to think of HIV and AIDS, how in the 80s HIV infection was lethal. And now this is something we live with, we manage. It's a very different situation. That's a virus. This is a degenerative condition. But I hope some of my message about boosting resilience shows that you can delay onset. If you delay onset, you change the disease. If you delay progression, you change the disease. So whether you will eliminate them entirely, maybe not, especially as we continue to age. But I think you can really transform, and I really believe this. I think you're not going to cure Alzheimer's. You're never going to put people back to their 20-year-old brains, and maybe you don't want to. But you will be able to completely. I mean, so these mice, when we restore their synapses, they get memory back, and that won't be all memory. But if you can do this with humans and just boost the sort of staying power of neurons, then I think you will transform these disorders. Long answer.
Thank you so much. Manuel, ¿nos podrías decir qué nos recomiendas para que vivamos durante más años y mejor?
Bueno, son recomendaciones muy generales y que están muy demostradas científicamente como por ejemplo, bueno, hay dos concretamente. Una es el ejercicio físico moderado. Esto está demostrado con estudios clínicos que mantiene el estado de salud durante más tiempo. Por ejemplo, el ejercicio físico moderado es tan bueno como la metformina como antidiabético. O sea, cuando se hacen los ensayos clínicos en diabetes, uno es metformina y otro es hacer ejercicio físico moderado, son igual de buenos. Y el otro es tener una dieta moderada también. La obesidad es un factor que disminuye mucho la salud en general. Produce multimorbilidad la obesidad, sobre todo en edad avanzada.
Muy bien, pues tomaremos estos consejos.
Suena fácil de hacer, pero luego es difícil.
Bueno, igualmente tomaremos estos consejos. Muchísimas gracias. Although we would love to hear more, we must come to an end as we want our people to also get to the next session of the Age Open Science cycle. The meeting is in the Palacio de la Salina in the Diputación de Salamanca. So I invite you all to continue your scientific journey through Salamanca. And a big thank you to our audience, the people that are following us on streaming, and to our speakers, Giovanna, Manuel. It has been a pleasure to have you. Thank you so much.
Thank you. Thank you.
Yes, we did.
This looks like big mess.